Okay, this mini lecture is going to be primarily about a protein called aquaporin. And aquaporin is found in all eukaryotes, and we'll learn more about eukaryotes when we get into chapter 6. The aquaporin story is really a story for chapter 7, and we're reading chapters a little bit out of order. We've got chapter 1, intro, 2, 3, 4, some chemistry, and then we built water, and we wanted to talk about a macromolecule, and our first macromolecule from chapter 5 was a phospholipid, like this, and we started to build a cell membrane. That made us want to go next to chapter 7 and learn about aquaporin and proteins in cell membranes. Cell membranes are composed of phospholipids plus proteins that are doing jobs in the cell membrane, like letting things in and out of the cell. So after we deal with chapter 7, we're going to want to go back to more of chapter 5 to learn about proteins in general. Aquaporin's a good one, and then we're going to head on to chapter 6, which is about all kinds of different proteins and membranes in cells. So that's where we're at. So we have just a little picture here of aquaporin. Aquaporin is protein. This is kind of like a cutaway of the membrane, and aquaporin looks like a donut. So water is going to diffuse in and out of the cell, and it's going to interact with the aquaporin protein through non-covalent interactions. So this will be a selective channel. It only lets water in and out of the cell, and it's going to allow water to diffuse rapidly. Okay, and where that's relevant is in this idea of the cell membrane as a semi-permeable membrane. Some things can cross the membrane and some things can't. And one thing that can cross this membrane, this is just an artificial phospholipid membrane or a cell membrane, is water. Water can go in and out of the cell. And it's doing that all the time. It's leaking in and out. And this is just stuff in your book about diffusion, and so this is where this topic comes up, is in the topic of diffusion and osmosis, water moving across the cell membrane. And these are important topics in the book, but they're covered really well in chapter 7. And so I made a osmosis handout to try to help explain some of the material in chapter 7. All right, awesome. And here's one of my favorite images, and that is that we can start out with just an artificial semi-permeable phospholipid bilayer separating these two open tubes. And if we have a higher concentration of a soluble molecule on one side than the other side, so a higher molarity over here, what will happen is over time, water will continue to diffuse both ways, but over time it'll diffuse in this direction at a higher rate, and as a result, the water levels will actually rise here and go down there as water moves here to cause this soluble molecule to diffuse, and that's going to continue until it reaches an equilibrium where the molarity on both sides of the membrane is the same. And it appears, that real work was done here, right? If you let this water flow downhill, you could use it to generate electricity like that. This is just like water behind a dam. And it's good to show you that. We'll talk about energy in chapters 8 and 9. We'll talk a lot about this idea that this higher concentration actually is like this. We haven't done any work here. We've just converted this stored energy to this. So having a high concentration of molecules on one side of the membrane versus the other is a kind of potential energy that's available to do work. And that's an important concept in biology that we'll get to. So diffusion, lots of different features of diffusion to be dealt with in the book and the textbook. Here's a key thing. Another diffusion and osmosis concept is that we can have a solution that has the same total concentration inside and out this side of the cell. That's isotonic. It can have a higher concentration outside. That's high outside hypertonic. It can have a lower concentration on the outside. That's hypotonic. And the book does a good job covering those topics. So all cell membranes have lots of proteins. So in addition to the phospholipids, there are lots of proteins floating the membrane, and that's where they do their job. And we're going to focus on aquaporin as one of them to introduce that and to introduce proteins in general. Here's another cross-sectional view of aquaporin. It's a water channel that shows that allows water to diffuse in and out of cells really fast. And the diffusion through the cell membrane of osmosis is not nearly fast enough to support the requirements of life. Say, for example, urine production in the kidney, tear production in the eyes have to produce big volumes of water going in and out of cells. So just to show you, this is a view of that donut. So we had this cross-sectional view, a little bit of a cutaway view here. And so here's the orange donut sliced. Here's the donut hole. And what's going on here, see, is this the cell membrane. And this shows another view of aquaporin where nothing is cut away, but instead the protein is sort of flattened out. It turns out that all proteins are chains of amino acids called the primary amino acid sequence. Remember from the first mini lecture recording or audio-video handout, the insulin protein is a long chain of amino acids of 20 different amino acids, and that's what these are here. These are amino acids with the single letter amino acid designation like methionine. And so this is aquaporin. Remember, it's somewhat floppy because covalent bonds can rotate around each other, and so we can have a sort of a cooked spaghetti. And the aquaporin goes in and out of the cell, and on the outside surface, these are probably generally hydrophilic amino acids, and these are probably generally hydrophilic amino acids, and then these are probably generally hydrophobic amino acids. If you draw that out, we'll see, like, there's the hole, and these tend to be a twisted ribbon, and so this donut is actually composed of a series of twisted ribbons, and it makes a sort of a barrel shape. And so on one side of each of these twisted ribbons, it would be hydrophobic, water-fearing, but then the donut hole must be hydrophilic, water-loving, to let water through. So this side of each of these chains must be hydrophilic. So one side of these twisted strands would be hydrophobic, and the other side would be hydrophilic. Then another thing is that aquaporin is a selective for water. So it's not just that it's a small hole that lets water through, because water's small, but like I said, there are specific non-covalent interactions, hydrogen bonds, non-covalent bonds between water and the amino acids in the donut hole, in the center of aquaporin. And highlighted here in blue are some of the most important amino acids that actually form hydrogen bonds with water as the water goes through. So here's the protein, and it looks like that, where to get through this small opening, the water has to make a non-covalent interaction. So that's some of the structure of aquaporin specifically, and we can start to see how structure and function are related. And then this is the structure of amino acid containing protein in general, long chain. Here's another view of aquaporin, just showing this idea that that long chain of primary amino acid sequence can be coiled up, shown as coiled coils, like coils, that create a barrel that water goes through the middle of. Here's just a kind of an interesting image of the, it's hard to see, but aquaporin protein in the cell membrane with water going through. It's a little bit hard to see here, but there are actually 4 aquaporins together here. So there are 4 different water channels. Here's an interesting view now where we've zoomed in on these amino acids that are especially important for water to make hydrogen bonds with as it passes through this restrictive point in the channel. So aquaporin creates a channel that lets water through, and only water, partly because of its physical size, but also partly because of its shape and its chemistry. It's polar and wants to form non-covalent hydrogen bonds. Okay, so it's a chain of amino acids chained together like that, and then these letters are specific amino acids that are important in the center core, like that. And there'll be a single letter designation, so this is amino acid arginine, and then a number, because the amino acids in a chain of a protein are labeled as the type of amino acid and also their position along the length of the chain. So aquaporin is a few hundred amino acids long, and along that long chain, there are some amino acids that are particularly important. And they're not right next to each other in the primary amino acid sequence because the protein folds up to make a three-dimensional shape. So we're moving toward understanding that about proteins, that proteins fold up to make a three-dimensional shape. So great. So we have these, and we'll also see that these are important. And remember we learned that when we eat food, whether in this case whether it be plants or animals, some of that food has aquaporin protein in it. We need the aquaporin protein, but we don't use the aquaporin from our food. Instead, the aquaporin in our food is broken down, and then the individual amino acids are chained together using the DNA code from humans to make human aquaporin. And there's a gene for aquaporin that is used to guide the chaining together of these amino acids. There could be a mutation in that chain, and the mutation might be here, an amino acid that's not very important, or it might be here. And you can get a first hint of this, that a person can inherit in their DNA a mutation defect that leads to the synthesis of a defective protein that can't do this job, and essentially we end up with a plugged channel. And we'll talk a lot about that. Okay, here's an example. Here's a mouse that has a mutation in an aquaporin gene, and when it grows up, it gets cataracts. Now, this is not the cause of cataracts or all cataracts in people, but is an example of a congenital, that means inherited, cataracts that formed in mice. And what's going on here is it's like the plant roots that we saw in an earlier mini lecture, where when the mouse has a mutation in it, the aquaporin is not able to flow in and out of the cell as efficiently. As a result, this mouse has dry eyes its whole life, and that leads to a premature onset of degrading of the lens. And so now we're starting to talk about proteins and the idea that proteins are made using the DNA as the code. The DNA we inherit from our parents, that DNA can be defective and lead to the production of a defective aquaporin protein, and that's true for any protein. Here's a picture. All these pictures, I'm just grabbing off the wind. Here's a picture. We're looking down the donut hole, and it's showing the waters passing down through the donut hole and some of the positions and types of amino acids around this chain, like that. And then check this out. Here it's saying that there can be a mutation in a gene called aquaporin 2 that leads to a special kind of diabetes called nephrogenic diabetes insipidus. And what's going on here is there's a mutation in the DNA that leads to a change where instead of arginine amino acid in the chain, there's cysteine added. You can see here that arginine is important in making a specific non-covalent bond with the water. So people can have that mutation and have that disease. Then here's another one that's interesting to look at. You can have a mutation in aquaporin 0. We'll talk about the different forms of aquaporin that leads to the cataracts, like we just looked at the mouse. That can happen in people too. And in this case, there's a mutation that leads to a change from glutamate to glycine. And this is interesting because, see, look, here's amino acid, and it's not in the core. It doesn't directly interact with water, but if it's mutant, there's a problem. There's a defect with the protein. What's going on here is it's showing that this protein actually makes non-covalent bonds with other amino acids that are essential to be properly positioned in the core hole, donut hole of aquaporin protein. And so this amino acid is important in stabilizing the structure of that channel to make it a stiff, physically stiff and specifically organized channel or hole. All right, great. So here's just some imagery. Aquaporin protein is like a donut, and it's actually like a raised donut because it's floating in the membrane. The browner parts of the donut are hydrophilic because they're exposed to the water environment outside the cell or inside the cell. And then see this belt of lighter material? That's hydrophobic amino acids because aquaporin is floating in a phospholipid bilayer, and this is that lighter region. It has like a belt of hydrophobic amino acids on it so that it can bob in the membrane. So we're starting to see some basis for the shape, the structure, and the function of the aquaporin protein. I guess I've written this all down here. And yeah, the whole point of this function is for water to fuse through here. The idea is that life in general, you know, has to be organized and specific. And one of the ways that gets done is to take the many, many different atoms in a protein, join them all together so that their interactions start to lead to a more stable protein structure and function. So in an environment that is otherwise pretty chaotic and random, proteins become highly organized and stable because of all of the covalent and non-covalent bonds. Here's back to this picture again. Again, emphasizing this idea that there are these specific connections get made, and mutations can lead to the production of proteins that are defective. And I think we've seen all these. Yep, so we're good. Good. And now here's the next thing, and that is in humans, we actually have 10 different aquaporin genes. So we inherited 10 versions from our mom and 10 versions of our dad. And why are there 10 versions? Because although they're all involved in water, osmosis, some of the aquaporins also allow some other small molecules into the cell, like that. So there's some specialized forms of aquaporin that are used in different locations for different functions, like that. So it's a nice list. Here's another way of drawing those 10 aquaporin proteins. Because they have, in their amino acid sequence, they have similarities to compared to the others. And so, for example, 5 and 2 aquaporin genes have the most similarities and appear as if they had a common ancestral gene most recently. On the other hand, these aquaporins are only distantly related to those. So a doubling of the gene might have occurred back here, and then the two genes evolved under separate pathways. Here's another example of that, just to try to connect it back with the idea in the first video lecture that insulin, a protein, was like a screwdriver. So here we have like the ancient aquaporin gene, and by chance there was a gene duplication event. That's a type of mutation where accidentally the DNA copies that part of the gene or that part of the genome twice, and so we get 2, and then later another duplication can have. And we're looking at screwdrivers, and so we're just looking at diversifying the screwdrivers, just like we're showing here, you know. Start out with one basic screwdriver, and then you're like, okay, well, yeah, actually, it would be nice to have both a standard head and a Phillips head. And then it'd be nice to have some really short-stemmed screwdrivers as well. So we have a term for that gene duplication product. So here's the single gene that got duplicated, and we now have two. One of them can perform the ancestral function, and the other can evolve somewhat to perform a new function. Stories of this type are called paralogs, where a paralog is two genes in the same species that share a common ancestral gene. And over time, they can become quite different. So here's Aquaporin, and here's this shovel. And that's not a screwdriver, but you can see over time it's evolved to attain a new function. And that happens really frequently in evolution, where gene duplication leads to a gene that makes a protein that plays that essential role, and then a new gene, a different gene. Here's just a fact about Aquaporin that's pretty surprising. We can draw pictures of the shape and structure and function, but it's amazing to see the rate of events that happen in biology. 1 billion water molecules can cross this membrane of one aquaporin protein per second. Humans haven't been able to invent a filter for water that's that efficient. Here's that image again, and then I think we're going to be in good shape with this. Oh, yep, and the next thing will be to talk about the specifics of the individual proteins and the amino acids that make them up. And so the next video will be about talking about aquaporin as an example of a protein in Chapter 5 and how the protein gets made.